Assalamu alaikum, sir.
Walaikum salam. Good morning. And how are you all? Tasneem, how are you?
Sir, I'm fine, sir. How are you, sir?
Fine, thank you. And yesterday, your friends, Kashfi, Oreen, and Rojana, I saw they were in hard work in the field experiment. Our wheat experiment is, in a peak time. So we need lots of people to work and get the, valuable data. Anyway, how is your health now?
Sir, there is some post-COVID weakness, but I think I will be able to join my mates soon,
Yeah.
in next week maybe.
All right, because we need to make some video clips of the current condition of the field. I advised them. I hope you can make a contribution there. Anyway, I welcome all of you to our course, Introduction to Ecological Chemistry. And do you have any questions from on previous lectures? If you have any question, you can ask, and otherwise I shall move on discussing the behavior of funny behavior, even geometric or mathematic behavior of the ant social insect. So any question? If no,
I
then
actually had a small one.
Yes, please.
Sir, in your previous lecture, you showed us some chemical structures of different pheromones. So my question is, do we have to learn all those structures for the exam?
Yeah, this is important question. Structure, in the world, chemical structures, maybe billions of structures discovered by human. All the structures are no need to memorize, but if you know, for example, if I ask you what was the first antibiotic discovered by human, you answer penicillin. That means you need to memorize some of the informations. So exactly not the chemical structure, but if I ask you to cite some examples of pheromones, and you should know some of the pheromones, at least the chemical name. Otherwise, understanding of the chemical ecology is not good, and it is better to have skill in drawing some of the structures, not all the structures. Some classical one, for example, the bombicol. If you ask to write something on pheromone, if you can answer with a name and chemical structure of a specific pheromone, then it would be a complete one. Is it clear?
Yes, sir.
Okay.
Thank you.
You're welcome. So I'm going to show you the behavior of ant, and behavior of ant also associated with chemical communications. I guess you can see the slide, right? Ant behavior
Yes, sir.
Naturally inspires scientists very much. Because not only scientists, even general people, and behavior is interesting. Because and behavior is thought to use to solve real life complex problems. By observing ant behavior, scientists have begun to understand their means of communication. Their means of communication means they use some mode or some technique or some language for their communication, and based behavioral patterns to address combinatorial problems first proposed by a scientist, Marco Dorigo. Here you can see very interesting image. Ants secrete pheromone while traveling from the nest to food and vice versa in order to communicate with one another to find the shortest path. It is very interesting. Ants are social insect and they are very hardworking insect. Every morning they move around to find the food source, and once they get the food source you can see how much weight they can carry. An ant can carry even 10 times heavier than their body weight, the item to their nest. And they are social insects. This is why they use uh, pheromone signal to uh, give the trail of their movement. Uh, but very interestingly is that here you can see in the image and travel, in different directions, but they can give the path, the shortest one for the other members. It is really interesting how they do that. More ants follow a trail, the more attractive that trail becomes for being followed. Here you can see people did some experiment, designing an experiment like this. So they put food here, here is the nest of ant. You see this path is the shortest and another one is the longer than this one, and this is the shorter. But ant didn't follow this path to reach the food source. They followed this way. Now question is how. It's really a big puzzle. People are surprised to see this. Here you can see some more experiments. The more ants follow a trail, the more attractive that trail becomes being followed. Very interesting. Nest, uh, nest, and food source. You see straight path they follow, and there no barrier was given in the experiment. But here a barrier is given. You see. Uh, if ant follow this obstacle and pass through this way, it would take longer path, but this one shorter. But what ant did, ant uses both path, and finally within a short period of time it was observed that no ant follow the longer path. That means the more ant follow the shorter path and more trail pheromone is there. And this is interesting. And the real sensory system are not clear, but it is already been discovered that they release some pheromone when traveling. The shortest path, the concentration of pheromone is higher. Now question is how they determine the concentration of the trail pheromone very quickly without moving the both path. But lots of question is there, but phenomena is very clear. Phenomenon is very clear that finally ant follow the shortest path to reach the food and get back from the food nest. How they select their route. Ants are forced to decide whether they should go left or right. First time they don't know. And the choice that is made is a random decision, not a precise for the first time. Pheromone accumulation is faster on the shortest path. The difference in pheromone content between the two paths over time makes the ants choose the shortest path. positive feedback mechanism to arrive at the shortest route while foraging. It is called stigmargy or stigmargetic model of communication. So many mathematician and many expert in modeling, they studied with ant a lot, and the model is called stigmargy or stigmargetic model. So you understood that first time they are forced to decide whether they should go left or right, but later uh, shorter distance path, um, higher accumulation of pheromone that help others, even the uh, pathfinder, to decide the shortest path. Different optimization problems have been explored using a stimulation of this real and behavior. And already the pheromone, this definition I have given you, but you can, for academic purpose, you can also go through this one. Pheromones are species-specific sensory input from the social environment that influence a definite behavior and developmental process. In case of ant, we have seen. And obviously, pheromones are active, uh, intraspecific. And here is the uh, uh, first description of pheromone given by Carlson and Lucia in 1959, which was published in Nature. So this is very interesting and seminal paper on the term of a kind of biologically active substances called pheromone. So I mentioned some of the semiochemicals like elomone or chiromone. What are elomone? Elomones. An elomone is any chemical substance produced and released by an individual of one species that affect the behavior of a member of another species to the benefit of the originator, but not the receiver. It is very, you should be very clear. In case of pheromone, pheromone is a chemical substance produced and released by an individual of a species that is, used by the members of the same species. But here, in case of elomone, is different. The producer is benefited, and producer is one species and the receiver is another species. For example, flower fragrance. Flower fragrance is, volatile compounds. They are released from the flowers, plant flowers, and perceived by the insect, for example, the pollinator. And ultimately, when insect comes to the flower and pollinate, the producer gets the benefit. So we can say those aromatic compounds, flower aroma or scent, they are elomone-like compound. Production of elomones is a common form of defense. Yes, in case of flower, aroma, they are not basically the defense, but in case of other elomones, they are basically defense molecules, particularly by plant species against insect herbivores. For example, bad odor in the plant. There are some plants which emit very strong and bad odor, and they are actually elomones to ward off the enemy, or ward off in case of plant herbivores. Herbivores means those loves to eat plant. Even mint plant use the strong aroma in the special cells on the surface, hairy structure. They are terpenoidal compound, uh, and they are basically used to deter or ward off the herbivores. Many insects have developed ways to defend against these plant defenses. Is an evolutionary arm race I mentioned before. One method of adapting to pheromones is to develop a positive reaction to them. The pheromone then becomes chiromone. This is very interesting. Uh, pheromone in the evolutionary arm race can be turned into chiromone. When it will be chiromone? When the target organism uh, uh, is adapted to the uh, defense molecule and use it as a clue to find the producer and use as how to say prey, then it is chiromone. Others alter the pheromones to form pheromones or other hormones. So in the evolutionary process, one organism when makes or synthesize a defense molecule against some of the other competing organisms, and if competing organisms somehow overcome the toxicity and use it as the benefit of them, then it is called chiromone, and it can be a pheromone of other organism, target organism also, or even hormone. Yet others adopt them into their own defense strategies. Sometimes a chemical produced by a plant is extracted by the herbivore, for example, insect larvae, and use them as their own defense, as I mentioned in case of butterfly, because in the larval stage they extract lots of toxin from the plants, and then they transfer them in the outer surface as a toxin signal, warning signal for their predator. For example, by regurgitating them when attacked by the insectivorous insect. So chiromone, as I mentioned, a new logism using the Greek word, which means opportune moment, paralleling pheromones, is a semiochemical. Obviously, pheromone, chiromone, both are semiochemical, emitted by an organism, which mediates interspecific interactions in a way that benefits an individual of another species. That means producer is not benefited by the semiochemical. Rather, the target organism overcome the toxicity as well as defense mechanism and use their own benefit, which receive it without benefiting the emitter. So emitter who is producing it not getting any benefit. This EBS dropping is often disadvantageous to the producer, though other benefits of the producing the substance may outweigh this cost. Hence, its persistence over evolutionary time. The chiromone improves the fitness of the recipient, and in this respect differs from an pheromone. But pheromone is beneficial for the producer, which is opposite. That means chiromone and pheromone, they are opposite in the benefit of the producer. And a cinnamon, which benefits both parties. There is another term, cinnamon. Cinnamon means a compound released from the an organism which benefits both producer and the receiver. For example, in case of flower, uh, fragrance benefits both plant as well as the pollinator. The term is mostly used in the field of entomology. Uh, Two main ecological cues are provided by pheromones. They generally either indicate a food source for the receiver or the presence of predator, the latter of which is less common or at least less studied. Mostly, the pheromone is used to find the a food source, but it can be also used to find the predator, locate the predator, but the later one studied poorly. I can show you one pheromone which we discovered when I was a student at Hokkaido University. The image, I guess you can see very carefully. The left hand side, a root tip when immersed into juice spore suspension. Perhaps you know the juice spore, Phytophthora, Pythium, Ephanomyces. They are fungus-like microorganisms. They infect the plant by using asexually produced biflagellated juice spore. And those juice spores can sense the host signal. Where is their host? They are very specific. A spinous root tip, when I immersed into the suspension of juice spore in a petri dish, you can see white dots. There are millions of juice spore aggregated at the root tip. And then they germinate and infect the plant. That means plant root release some signaling molecules that are guided the pathogenic juice spore to find the host. That means the chemical produces by the plant are not beneficial for them, rather the harmful, because pathogen using this chemical clue to find the host. It was a hypothesis. The process is called chemotaxis. Chemotaxis means in ecological chemistry, a nice term, preferential movement of an organism toward a chemical source, chemical attractant. So chemotaxis was a phenomena observed by many scientists. During my young career at Hokkaido University, I was asked to discover this chemical signal. What is the signal released from the root of a host plant of Ephanomyces cochleoides, which causes damping of disease in sugar beets, spinach, and some other members of Canopodiaceae and Amaranthraceae, to discover that compound. So first we observed the true chemotaxis, that is when you put a root tip into juice spore suspension and observe under a light microscope, you can see within a minute millions of juice spore attracted and come to the root surface, and they shed their flagella and started germination and penetration. And in some part, right hand side, you see juice spore produce mass of cells, and the shed flagella become cystospore to make the inoculum potential for infecting the root. Our question was, what is the signaling compound? Here you can see cochleophilin A. We named our discovered compound cochleophilin A. By using a series of chromatographic process, we finally discovered this flavone signal, and this signal is incredibly powerful in attracting the juice spore. And when juice spore comes, this is a bioassay method we devise because it happens in the rhizosphere root zone in the soil, so you cannot see. To see the behavior of juice spore, we produce juice spore in the petri dish. There is a long process, and we devise a method called particle method. I would show you later on. And what we did, the root extract in ethyl acetate solution, absorbed by this black part, means a porous particle used in gas chromatography around 200 micrometer in size. And when you put this particle with the solution of root extract, and ethyl acetate easily evaporate, and the chemical remain in the porous particle. When you drop the particle into the juice spore suspension, here you can see lots of spores. It feels that it is a root because it produces a gradient of compound slowly diffuse in the water and attract them. And all juice spore, when comes to the particle, they become cystospore by shedding their flagella and germinated. Even cystospore germinated a little far, they show the chemotaxis or germ tube tropism. Here is the scanning electron micrograph. This is not root, actually a particle coated with the cochleofilling A, and it was used like a dummy. So juice spore thought that it is a root because they are getting the same signal they receive from the root. So and the right hand side is the real root. In real root scanning electron micrograph, you see lots of spores germinated and penetrated to initiate the infection. So cochleofilling A discovery was very amazing, because before that discovery, many scientists published dozens of papers that thought different ions or pAs or other things are involved in the chemotaxis of juice spore to the root. But we discovered a genuine ecochemical which is specific for the Pheromyces cochlearis juice spore. Other juice spore never respond to this one. And this compound only biosynthesized by the host of Pheromyces cochlearis. That means this signal ring compound is very specific for the attraction of Pheromyces cochlearis, and it is a true pheromone. And you may ask question, why host plants like spinach, sugar beet produces cochleofilling A, because it is a harmful compound, invites the pathogen and help pathogen to find the plant. Possibly this flavone was biosynthesized as a defense molecule, but over the years of evolutionary process, Pheromyces cochlearis developed mechanism to perceive this signal at low concentration, and they adapted to its toxicity. So it is in Pheromyces cochlearis, in our laboratory, we found that it is not at all toxic, rather helpful for their growth and development. The real mechanism we don't know, but we discovered the receptor protein in the juice spore membrane, and we found the signal transduction pathway, because this molecule at higher concentration trigger expression of certain gene to induce juice spore to cystospore, because juice spore cannot infect the plant. It needs to become cystospore and germinate and form the germ tube. I shall discuss later on this story. And this is a fascinating story so far discovered in the field of chemical ecology, how a pheromone signal is used by the pathogen to locate its host plant, and then the same molecule how at higher concentration at the root surface use to morphological changes of the pathogen to successfully initiate the infection. And you may ask the question, what is the use of this discovery? This discovery, this is a fundamental and
Sir,
yes,
sir,
yes.
then according to the rules of evolution, shouldn't the plant also evolve accordingly and stop producing such chemicals which attract harmful microorganisms?
This is interesting question, but there is no evidence that in case of pheromone, the plant stops or the producer stops this, producing this compound. There is no such evidence because in biosynthesis of cochleophyllin A, there is a pathway in the cell of the host. But it is a really nice question. If someone can find that a pheromone over the years of evolutionary process, when the producer sense that it is harmful or it is used by the enemy, then the producer stops to synthesize it. It is a very interesting question, but in case of evolutionary process, it is not in one day. We do research, 5 years, 10 years, or 20 years like that. But this evolutionary adaptation probably happened by several thousand years or 100 years or like that. But your question is obviously valid, and for the fitness of every organism, if they sense that it is being used by the enemy as a clue to find the producer, they may take decision to stop synthesizing it. But I couldn't find any literature related to this. What plant usually do in the coevolutionary arm race, when cochleophyllin A-like compound are no more toxic for the target organism, what they do, they go for synthesizing more toxic compound. This is why in the plant, if you analyze any plant, you can find thousands of, molecules. That means first one, when ineffective, like your version, in the arms, first version, once ineffective, then they go for the second version or third version or fourth version, which is called co-evolutionary arm race. But first version, second version also remain, not, stop the synthesizing. This is why, for example, in case of a medicinal plant neem, Azadirachta indica, scientists discovered 2,500 different secondary metabolites biosynthesized by Azadirachta indica, one of the richest source of secondary metabolites. Do you know why? Because cochleophyllin A may be ineffective in stopping Phenomyces cochleoides, but this compound was not specific. All constituent defense molecules are not specific for particular organism. It might be effective in protecting other organisms, not only the Phenomyces cochleoides. This is why they keep it, because all organisms are not targeting the a specific plant, so they have the also specificity. This is very interesting point. For example, tobacco plant. Tobacco plant biosynthesize nicotine, very toxic alkaloid. in their cell. Most of the insects, even human, avoid the nicotine because nicotine is a neurotoxin. But one insect, Menduca sexta, their caterpillar loves to eat tobacco. Do you know why? Because here competition is almost nil. Other insects are not competing of getting the tobacco leaves. So over the years, they adopted a very interesting, funny mechanism. What they do when they are feeding on tobacco leaves, they extract nicotine and purify nicotine and drops the pure nicotine from outside the body, just like a liquid drop or like that. And if you remove the nicotine from the leaves, other parts are highly nutritious. So these are some sort of co-evolutionary, arm race, and very interesting for the practical application. For example, in case of cochlear A, when we discovered it, scientists and industry in Canada, they use the cochlear A as a bait in the fungicide or pesticide. So juice-puric organic Aphanomyces cochlearis feels that fungicide particle, they do not know the poison over there. As cochlear A is a hydrophobic compound, slowly diffuse, attract the juice spore. When juice spore attract to the source of cochlear A, they find the toxin, and that is the way of attracting the pathogen, and then smartly killing them. And this is the practical application. There are lots of practical applications. For example, if you engineer the sugar beet and spinach by silencing the gene involved in cochlear A biosynthesis, you can develop a variety which would not be, sensitive to the damping of disease caused by the Aphanomyces cochlearis. So this is all for today's talk, and I think you can ask some question.
Sir, I have a question regarding the ants behavior part.
Yes, sure.
Sir, I didn't really understand the positive feedback mechanism point.
Positive feedback mechanism of which one?
Of ants, sir, when one ant follow another ant's trail, and how they decide which trail to follow.
Yeah. Actually, when an ant travel shorter distance to find the food, and then the trail pheromone concentration would be higher and compared to the longer distance. So, and usually I told you, they do experiment like this way. Initially, both path they use. You see, in the obstacle, this experiment was done in the sea. So both path is followed, but to do some trial, because initially they cannot decide. But when shortest path is followed, the trail pheromone concentration would be higher than the longest path. As a result, they have the sensory transduction system to discriminate the concentration of the trail pheromone on the path. So finally, in the D, you can see all insect, that is ant, follow the shortest path. So there might be some more other interesting mechanism that follow by the ant, but so far The current understanding is that the concentration of the trail pheromone when it travels shorter distance on the path is higher than the when they travel the longer distance, because total trail pheromone biosynthesis is not unlimited. This is why they use in the shorter path higher concentration than the longer path, and they have the mechanism, unknown mechanism to discriminate the concentration. Minute differences in the these are femto molar concentration or like that. They're so smart.
Sir, I have a supplemental point.
Yes.
Yes, sir. Yeah, sir, does each of the ants producing the same pheromone, or some are producing and some are just following the trails?
This is interesting. Ant is a social insect. There are some social insect like honeybee or a termite. They have different class, working class, search for food. And there are, some males or the queen or like that. So actually, the workers hunt for the food, and they are, they need the trail of the path, shortest path to find the food, because longer path, if they travel, they need more energy. This is why I guess not all ant uses the same trail pheromone. Workers for finding the shortest path, they use the pheromone. Maybe not a single pheromone, lots of other pheromones for other purposes. The ant and other social organism may use. Some of them are defense, warning pheromone, as formic acid produced by formic acid and some other acid, toxic acid, produces by the ant. And when they bite, you can feel pain or like that. So lots of ecochemicals, not only a single one, are produced by the insects. But initially, maybe in the starting of the evolutionary process, there were not so competition. At that time, probably no such a secondary metabolism was required. But day by day, the system becomes complicated, and lots of other, competitors and stressors develop to find the for their fitness, that is to overcome the stress, as well as for their own benefit or communication. Organisms through the evolutionary process are becoming complex, and more new ecochemicals are required to sustain or for their fitness in the changing environment. Is it clear, Amim?
Yes, sir. Yes, sir. Thank you, sir.
Welcome.
Sir, if we spray another perfume, will the ants get distracted?
If you spray the trail pheromone, yes, it is possible if you know the structure. And if you spray the trail pheromone around the ant, they will be misguided, because chemicals either synthesized by the organism or prepared in the laboratory. If you know the structure, if it is structurally identical, it does not mean whether it is from the natural source or it is from the artificial source. Many people comment that you take amloki to get the vitamin C is beneficial than getting L-ascorbic acid as tablet or like that. But in science, there is no difference because chemically is same. But when you take amloki, maybe you are getting other nutrients or minerals or like that. That would be beneficial. But pure one, either it is synthetic or natural, the function must be same. This is why you may use the trail pheromone to misguide the and you can find some literature, maybe some people did such funny experiment. Good question.
Thank you, sir. And sir, can you please repeat the term chemotaxis?
Yes, chemotaxis. This is interesting term. Preferential movement of an organism toward a chemical source. For example, cochleophyllin A. Cochleophyllin A, through the evolutionary process, is a pheromone for Aphanomyces cochleoides juice spores. So they have the receptor and sensory transduction pathway no other organism have. So when cochleophyllin A is in the environment, juice spore can sense and they move toward the source of the cochleophyllin A. And such movement is called chemotaxis. That means preferential movement of an organism by a chemical. People use different aromatic compounds, perfume, and it is also, how to say, some sort of attractant, or people feel good when they sense, and chemical, sensing of different organisms to chemical really very exciting. For example, chemotaxis of insect toward particular flower aroma. Later on, you can understand all flowers are not visited by all insects. They have the specificity. So a chemotaxis is like that. Even pheromone. Pheromone is a chemoattractant. So particular insect, they can sense and move toward the source of the pheromone, because when they in the source, their concentration is higher and they diffuse through the environment and gradually concentration decrease. So the chemotaxis means follow the gradient from lower concentration to higher concentration. This type of addicted movement or like that. Clear?
Yes, sir. Thank you, sir.
Sir, I have a question.
Sure, Rojana.
The question is that a particular insect always secrete the definite amount of chemical, or always, or depending on the situation, this amount may differ.
Good question. Yes, depending on the environment, everything can be changed. As you are a student of biotechnology, depending on the environment, gene expression is regulated. An organism may have a genetic makeup, but environment nearly regulates 50% of the expression of the gene. So genetic makeup cannot give you guarantee that you would get the real phenotype in any environment. For example, a plant variety, if it is susceptible to salinity, but it is very high yielding variety. So if you grow in the saline environment, some of the growth related genes would not express and you would not get the expected yield. So biosynthesis of compound also depend on temperature, depend on weather, depend on the age of the organism, depend on many factors. So not necessarily, for example, you see flower aroma. When flower blossom, then you can get the aroma. Although the genes are present in all the cells constitutively, but during the need of getting the pollinator, the aromatics substances synthesized. That means particular genes involved in synthesis of those molecules expressed due to the particular age of the organism. So age is very important. Environment obviously very important because environment regulates a larger proportion of the gene expression. So gene expression is the key for synthesis of particular molecules. So all the molecules synthesized in the cells of any organisms, there is a pathway, multi-step pathway. Lots of enzymes are involved, and those enzymes are through transcription and translation process, they are synthesized at the ribosome, and this particular gene expression taken place. MRNA comes from the uh, nucleus to the ribosome, and then synthesize that particular protein. And some proteins are enzymes, and those enzymes participate in the biosynthetic pathway of the molecules. And one thing you must remember, that all the molecules, ecochemicals, they are mostly secondary metabolites, and secondary metabolites, they are derived from the primary metabolite. Secondary metabolites are not directly involved in growth and development and reproduction of the organisms, but they are very important as we are discussing. Primary metabolites, for example, protein, amino acid, sugar, they are directly involved in growth and development. But for the synthesis of secondary metabolites, for example, an amino acid can be used to synthesize an alkaloid, or a carbon containing compound, which is a primary metabolite, can be used to synthesize a phenol or a flavonoid or others. We shall discuss the biosynthetic pathway of the molecules later on, but now at this moment, you need to understand that all the ecochemicals, they are derived from the primary metabolites in the cell by or through a specific biosynthetic pathway. And it is expensive to synthesize the molecule. This is why plants do not, or even any organism, do not synthesize any molecule unnecessarily. There might be some meaning for their fitness, associated with their fitness to the environment. So sometimes environment forced to synthesize more particular compound at larger quantity. For example, in a country, in the normal condition, the ammunitions in the army or defense forces required, or when the country is in war, the requirement is changed. So similarly, when organisms are in a particular stage of life, or under a stressful environment, they biosynthesize molecules at higher quantities, and that means environment trigger expression, higher level of expression of specific genes for synthesis of the molecules. Very interesting question. Clear?
Sir, then what in case of ant, we say that the trail pheromone is limited. Then if the synthesis of the chemical is may vary with situation, then when they are passing through a long distance, why don't they produce much more pheromone in that case of ant, sir?
Yes, of course. If they do not find the food, and if the food source is far, obviously they produce higher quantity of the pheromone. But synthesis of a particular chemical substance in the cell is also expensive. They need to have a balance because they need energy for their growth and development and reproduction. They need energy for biosynthesis of this type of compounds. So they need to have a balance. This is why, and you can see they always store some food for their future use so that they go, no need to travel so long always. But anyway, there are some, how to say, a balance. And according to need, they need to synthesize. Sometimes, if you sometimes for defense, higher cost may be higher energy may be invested for their survival or fitness. Any more question?
Sir, I have another question actually.
Sure.
Sir, we know that the particular insect species produce the particular compound, chemical compound, but what's about among the member of the same species? Like when a member secretes the sex pheromone to the others, then how others find that particular member? Is there any difference in the chemical type among the member of the same species to identify that particular member?
Member of the same species may use the same receptor because no need for communication among themselves, no need to use more energy. More receptor, more diverse compounds requires more energy. So in the same species, for exchanging same signal, all members may have same receptor and they use same signal. But in the language, as I told you, chemicals are the language of communication. So same chemical may not give us lots of language, lots of stories. So they may use different signal for different different compounds for different signals. Obviously, among the same species, depending on the environment, in different region, you may find structurally slightly different compound. For example, a particular organism in Bangladesh species and in Africa or desert in Arab, they may use slightly different structure, but fundamentally mechanism is same. So there are obviously in the same species, there are huge diversity. For example, rice. Rice in the Bridging Bank, they have more than 7,000 different, phenotypically different genotypes. All are, Oryza sativa, but they are phenotypically different. That means they are obviously chemically also different. So there are changes. But for example, Kalizira rice. Kalizira rice all over the country or in a field, all the plant may produce same aroma, not every spikelet producing a different aroma or like that. Is it clear, or do you have more question?
So actually, my question was that how the other insect in a same species identify the particular member. Is there any system?
Yes. They can identify the particular member. They have the sensory, as we have the sensory system. We can see even insect can also see. They can discriminate from others, and they can also, I told you, they can use the chemical signal, and chemical signal in the same species, they have the receptor, specific receptor. Other species have not, so they cannot sense. So there are lots of ways the organism can, sense and discriminate who is their community member in the social insect and who is not their community member. In Bangladesh, if someone comes from India, for example, West Bengal, difficult to discriminate. What you need to do, you can ask, show me your ID, national ID, or how to say, passport or like that to discriminate. But if someone comes from the Belgium or Norway, you can easily understand this guy is not Bangladeshi, because just to see or like that. So organisms are smarter than our identification system.
Okay, sir. Thank you. Sir, I have a request another. Actually, we have a request another that the recording of previous class was not given. So, sir, if you provide us, that will be helpful for us.
I shared with you through the, your Facebook group. And it is available in my, website. Okay, I shall share it again if it is missing. Any more question?
Sir, I have a question.
Yeah.
So we use pheromone for building pheromone traps. So can we use the trail hormone to keep the insects away from our field or lead them towards a trap?
Yes.
Can we use trail hormone in this field?
Yes, a trail, hormone, also pheromone also you can use. But the problem is, in case of a trail, how can you make the trail? Because when insect walk, the concentration at the mark, that means in their footprint, is really unknown. Very, very trace, and it is difficult to make such a trail, and they are not basically volatile, so volatile like pheromone or like that. But the idea is interesting. If a trail pheromone, if we can make something mimic the natural trail, then surely you can misguide the insect to a trap.
Okay. Sir, I have a request too, sir.
Yes,
Sir, can we have the slides of the class, please?
obviously. Obviously, we will get all the sides.
Sir,
I
thank
shall,
you, sir.
I shall share with you through your Facebook group. Good question. Any more? Naz, are you attending the class?
Yes, sir.
Any question from you?
No, sir. No, sir.
You understood everything? This is the, in my class, I always teach students not to memorize the thing, what I am telling, but to stimulate them or train them how to ask a question. How to ask a question is a very big skill. In a big international meeting or any meeting, even in your official meeting, if you can ask an interesting or intelligent question, then you can draw the attention and you can tell the society that how intelligent or learned or capable you are. So this skill would never come only memorizing and getting the highest grade in the exam. It needs some practice. First time, maybe your question is, some stupid or foolish or silly question, does not matter. So the more you ask question, the more you would be the intelligent, and you can have over the period of time, you can gain a skill. So I encourage students to ask the question, any type of question. Maybe your question is not answerable by me, maybe due to my poor knowledge, does not matter. And classroom is the safest place for asking any question, because you are a student. And when you go to office or anywhere as a graduate, then it would be challenging for you to ask a question, because people will evaluate your status or other things from your question. So do exercise in the classroom as much as possible. And master's degree is, the stage where the last stage of you to have this habit. Any question? If no question, then I shall close today's session, and I shall share the video as well as the lecture files, and wish you have a good day. Thank you.